Okay, now we're going into chapter 7. When you're doing research, as it says up on top, you're going to put together references. And part of the process is making sure no one has already written your thesis. You want to see what's out there and see whether your idea was had and completed by somebody else. Down at the bottom, it says total printed or photocopied, 248. And then it's got it split up. Papers found in the online database, so this is the stuff that we would have. Papers found from other papers reference lists. So as you read papers and they have references to, okay, we did this according to this process, or so-and-so had said this, then you need to backtrack and look at their references. And at least, and here's the thing, at least read the abstracts. This is the purpose of abstracts. You have a lot of stuff to go through, and you just will never have time to read every single paper, and every single paper won't be useful to you. So if the abstract is a good abstract, you can go through that one paragraph and go, okay, I need to print this out and read it. Or, nope, they never did what I thought maybe they did. I mean, with me, I was looking at particular cells and particular parts of a roundworm, and going through abstracts, you could, okay, they looked at something else, or they never found this kind of structure. Paper your advisor wrote 10 years ago, you've got people running your lab, probably in the same field as you are, you gotta be looking at their papers. Papers your advisor had forgotten to tell you about, that's gonna happen. So total printed or photocopied, 248. Papers actually read, 107. Papers actually understood, 5. Papers actually relevant, 2. Papers included in thesis reference list, 246. So when you do research, there's a lot of backstory that you've got to put together. You've got to figure out what's going on. And as we go through the semester, I'll use a lot of these particular cartoons, because if you're a science major, then most science majors wind up going on to beyond a bachelor's degree. And if you're doing beyond a bachelor's degree in science, you're doing research. So we talked about this already, but we're going to pick it up a little bit more now. ATP, adenosine triphosphate. This is, when it breaks apart, very exergonic. And I mentioned this when I talked about it before. Breaking that bond does release energy that can be transferred. But moving a phosphate onto another molecule can destabilize the molecule and supply the energy you need to get the molecule to change. That is called group transfer potential. And that phosphate on the end of the string has a very high group transfer potential. It can really cause stuff to happen. You put it together from adenosine diphosphate, and phosphate is represented a bunch of different ways. PO4 plus, is it P a little I, P inside a ring. There's a bunch of different ways that, depending on the what you're looking at, will represent phosphate. There's also adenosine monophosphate, where there's only one phosphate on the string. That's not typically used in energy reactions, but depending on how it's constructed, it can be a very key molecule in moving messages around inside a cell. So that'll come up later when we're talking about cell responses to messages. This is a chapter about energy reactions and energy relationships in cells. And we talked about how cofactors very often help in moving electrons around. Well, a lot of coenzymes also help enzymes in being able to mobilize electrons. Okay. Good. NAD. You can see why it's called NAD, because it's nicotinamide adenine dinucleotide. And I had to double check to make sure that I was getting it all right. It doesn't have nicotine in it. I've talked to a few chemists about it, and they're not entirely sure why it's in the name. But nicotine solutions were used a lot in extraction processes, and they think that might have to do with where the name came from. So it looks like it's got nicotine in it, but it does not. This is also a vitamin, right? Vitamin called niacin. It is B3. The B vitamins very typically are in this group of electron transferring coenzymes that move energy around. This is why you typically find things like Red Bull charged with B vitamins. Is that you make them, but we juice you full of them, then you'll have a few more to carry energy around. And you'll notice that they come out. If you've taken kind of a walk through respiration pathways before, you probably didn't really see this stuff involved, but as we walk through it, you're going to see it more. This is very often in redox reactions. Reduction, oxidation, electron transfers. Here's another situation where you have a pair of terms. One of them, because it's using an English word, is pretty easy to remember. And then you remember the other part of the pair and figure it out from there. If you move electrons to something, you're moving a negative charged electron to something that's now got one more negative charge than it did. Its overall charge is reduced. Ta-da, reduction. So a situation where a particle gains electrons, that particle has been reduced. The other particle that's lost electrons has been oxidized, because when people were first working out this chemistry, this is something that oxygen's pretty good at. So eventually, after they named it oxidation, they kind of generalized the process. So it doesn't just involve oxygen. It's any time that something loses an electron in a process, it's oxidation. Okay, here's another really important fact. Let me put this up there and I'll come back to it. If you're going to move electrons around, typically they're carried on hydrogen atoms and hydrogen ions. If you put an electron on a hydrogen ion, you have a hydrogen atom. So you start with NAD, you put H2 in there, you wind up with NHDH2 plus because only there's only one electron zipping around these two hydrogens. And then the hydrogen comes off and the electron has been transferred. Typically, these things don't show electron transfers. You just have to know what's going on. NAD is picking up an electron in the process, so it is an oxidizing agent. It's picking up the electron, it's reducing, but it's grabbing the electron from somewhere else, so it's an oxidizing agent. NADH, because it's contributing an electron, is a reducing agent. There is a variant that shows up in photosynthetic processes that's NADPH, just has a phosphorus in it. FAD, flavin adenine dinucleotide, is another one. It's also considered a vitamin, riboflavin, or B2. It's a little bit more of a straight carrier. The hydrogens are going to pull some electrons with it. It's going to move the hydrogens around. So FAD plus H2 gives you FADH2, and it takes the hydrogens usually to some place where they're going to be run through a membrane-bound processor and generate a bunch of ATPs. That'll come up later as well. So in this case, what's generating the process is electrons. So you're talking about electrical potential. These things have a high electrical potential because they have a high capability of moving electrons and feeding them into things. Okay, here's some old ideas. Remember I said we'd go back over stuff we'd already gone back over? In food chains, the producers, if you remember, are capable of taking energy from the environment and transferring it or converting it into molecular bond energy. They take carbon compounds, carbon dioxide generally, and take some sort of energy from the environment and use it to stick the carbons together and produce glucose. Not always, but most of the time. It can the more primitive ones, probably the original ones, were chemosynthetic. Chemosynthesis uses essentially heat energy to feed the process. So these are things that happen at mostly hydrothermal vents, cracks in the bottom of the ocean floor, where magma from underneath is mixing with water and heating it up. And then you have all these chemosynthetic organisms that are using the juiced, the heated up compounds to generate glucose. And then you have photosynthesis that uses some frequencies of light energy to stick the carbons together. So the light energy becomes bond energy, the heat energy becomes bond energy. And they're the bottom of the food chain. They're producing the fuel that everything else, including themselves, runs on. So then you've got the consumers. And there are levels to this, but it gets very, very muddy. You know, a primary consumer is something that would only eat producers. A secondary consumer would eat primary consumers. But, you know, there's so much crossover. Is a food chain really becomes a food web. You do have some organisms that are pure primary consumers, but once you get a next level up, it gets very, I mean, we're both primary consumers and secondary and tertiary and quaternary, is that we're consumers on a whole bunch of different levels depending on what food web we're pulling our food out of. And even things that we tend to think of as being like one type of consumer, like you go, oh, those birds are eating seeds. They're primary consumers. Well, I'm sorry, they're eating bugs too. So they're not just primary consumers. The number of niches, the number of potential jobs for producers is limited. Is that they need to be able to access the energy, they need to be able to fit into the room. And so they don't have the diversity of different species that the consumers do. But there's the mass of organic material, the mass of living material that the producers produce outweighs all of those different types of consumers. So more biomass on the producer side, more diversity on the consumer side. It's just a matter of, you know, once you get the food made, there's many, many, many different jobs in how you live and where you live and what you eat and who you eat and all that other stuff. So there's much more diversity on the consumer side, but they're working off of biomass that, for the most part, doesn't really feed up the food chain. So the biomass is kind of limited. And remember, at every transfer, energy gets lost and mass gets frittered away. Speaking of which, specialty consumers. They are consumers, but they specialize in material recycling more so than, I mean, we do a certain amount of material recycling. Right now I'm breathing carbon dioxide out, so I am recycling some of the carbon compounds that I've taken in as carbon dioxide, which plants will use to rebuild glucose. But certain types of bacteria that are involved in waste and dead thing breakdown, certain types of funguses that do the same thing, even some insects, because they specialize in producing a lot of raw materials from stuff that's kind of on its way anyway, they're considered to be decomposers, and they're kind of considered to be at the end of the food chain. Now keep in mind, every time you take in something and break the molecule apart, you use some of the energy, but some of the energy gets lost. So energy doesn't recycle back to the producers. Materials recycle back to the materials, but not energy. And in fact, for the second law of thermodynamics, a system like ours would grind to a halt. Some people go, oh, we break the second law of thermodynamics. No, we don't. Second law of thermodynamics says that systems will degenerate into entropy, into chaos, unless there's a huge energy input at the beginning, energy that can be used to produce order in the chaos. And of course, that big yellow ball up in the sky is our energy input. And for the few hydrothermal communities in the world, that's supplying energy for those particular communities. They're just few and far between. Okay, good place to stop for this part of the chapter.